Hey, this is Ralph from Pathognomonic, and welcome to my boards review crash course in immunology. These videos are intended for medical students preparing for USMLE Step 1, but really could be of use to anyone looking for a basic solid understanding of the immune system and how it works. If you have not already, please make sure to check out the video on this channel titled What is Pathognomonic, which will give you a sense of my approach to boards prep, why I think these videos can be helpful, and how to get the most out of them. With that said, immunology is a tough subject because there are a lot of details that you guys are expected to memorize and have already forgotten by the time you start studying for Step 1. It's also not an easy topic to teach because there are a lot of moving parts that are interdependent on one another, and it's kind of hard to know where to begin. This is why I thought it was a good idea to start with this overview lecture that will broadly cover the entire immune system to put into context where everything belongs, and also give you some more detailed background on certain concepts that are important to understand before we get into the lectures that follow. Let's first get into the main division in our immune system, innate and adaptive immunity. When we talk about innate immunity, we mean any part of our immune system which is inherent in our genes, so we're born with it and pass it on to our offspring, and it doesn't change or adapt over our lifetime. The things that contribute to innate immunity are physical barriers, like skin and mucous membranes, which includes gut and respiratory mucosa, and the defense cells at these superficial levels. Basically, anything that is exposed to the outside environment must have innate defenses. Proteins and chemicals are also used to fight infection. Things like cellular enzymes, complement proteins, which we will discuss in depth later on, and also acute phase reactants. Acute phase reactants are proteins made by the liver in acute inflammation. The key thing to know about innate immunity is that it's basically one size fits all, and there's not a whole lot of specificity to the response. But the upside is that it's there ready to defend right away and can recognize most common pathogens. If the innate defense cells do come across something uncommon or an infection gets out of control, they can alert the adaptive system. The adaptive system can handle the more complex stuff, but still relies on the innate system to signal that there's a problem. You also have to know that anything that is handled by just the innate system cannot contribute to immunologic memory. For instance, if you've ever wondered why you don't get a reaction the second time you have a PPD placed, it's because the protein is handled by the innate system alone without triggering adaptive immunity. In contrast, the parts of the immune system known as adaptive immunity are acquired, and these types of microbial resistance cannot be inherited. We are born with their potential, and over a lifetime, the immune system adapts and gets better at fighting infection. The workhorses of the adaptive system are T cells and B cells, and the main job of B cells is to make antibody, which can be viewed as the end goal of the immune system. Compared to innate immunity, the adaptive system takes longer to get going in response to an infection, but it will lead to a directed response toward a specific invading pathogen, and overall ends up being much more powerful. Just keep in mind that once adaptive immunity kicks in, it's not like it just takes over. Really, it supports our innate defenses and in many ways makes them better, and you're going to see this is a recurring theme throughout the course. And probably the most important aspect of the adaptive system is that it results in immune memory for T and B cells. These memory cells let the immune system respond more quickly and more powerfully the second time we're exposed to the same pathogen, and that's really what gives us immunity against certain diseases. A discussion of immunology is really a discussion of different immune cells and their interactions, so I think it would be helpful to talk about the lineage of these cells and how they relate to one another. Multipotent hematopoietic stem cells in bone marrow give rise to all blood cell lines in the body and are CD34 positive. Cluster of differentiation, or CD molecules, are surface markers on cells that have various functions, one of which is to help us identify cells in a lab using flow cytometry, and you know it's a stem cell if it expresses CD34. And from these stem cells we get both myeloid and lymphoid progenitor cells. When we go down the myeloid lineage, we see that this line gives rise to megakaryocytes, these large cells that produce platelets from their membrane, erythrocytes, which are red blood cells, but the one we really care about for our purposes is the myeloblast. Myeloblasts form immune cells called granulocytes, which basically means they contain secretory granules, and this includes basophils, neutrophils, and eosinophils. and also derived from myeloid progenitors are monocytes, which go on to form macrophages in tissue. On the other side of the cell lineage tree, lymphoid stem cells form natural killer cells and T and B lymphoblasts, which go on to mature into T and B lymphocytes. Just by the way, don't confuse lymphocyte with the term leukocyte, which just means white blood cell. Leuko means white, and all the immune cells we just talked about are types of white blood cells. Once mature, T cells can differentiate into either cytotoxic or helper T cells, while B cells turn into antibody-producing plasma cells. Some more terminology I want to clear up, which some people tend to get confused by, is that there's another arbitrary classification that's made based on cell nuclear structure. All of the granulocytes happen to have a segmented nucleus, so the term mononuclear cell is used to refer to any white blood cell with a nucleus that is not segmented, and this not only includes lymphocytes, but monocytes and macrophages as well. When you see the phrase mononuclear infiltrate, it's usually referring to chronically inflamed tissue filled with lymphocytes and macrophages. So now that we've seen the origin of immune cells, let's talk about some of the cells in the innate system that are always there protecting us from infection. Cells of the innate system have a fairly primitive way of killing invaders. They eat them and digest them. This process is called phagocytosis. The way this works is that the cell extends these long arm-like projections called pseudopods to envelop whatever they think is threatening, and it ends up in a vesicle inside them called the phagosome, as is beautifully demonstrated on this slide over here. This phagosome can then fuse with a lysosome, a body within the cell that contains catabolic enzymes, and together they form a phagolysosome, and this causes the ingested material to break down. There are a few cells in the innate system that specialize in this, which are called phagocytes. The first are the macrophages. The literal definition of macrophage is large eater. These cells live a long time in tissues and are usually in resting phase, meaning they just hang around eating random stuff until they find something bad. Part of this responsibility includes cleaning up cellular debris, and cells like this are sometimes referred to as sentinel cells, because while performing their other tasks, they can react if they encounter something harmful, such as this macrophage, which has just swallowed some kind of fungal cell and appears to be in the middle of mitosis. Macrophages release cytokines, cell chemicals, to recruit other cells and trigger the inflammatory response. They are our first line of defense. One of the cells that can be recruited to this scene is the neutrophil. Neutrophils can also kill by phagocytosis, but are capable of a much greater response because they can also release destructive enzymes into the tissue. This can very quickly do away with a large-scale invasion, but because this type of counterattack is non-discriminatory, it leaves behind some degree of devastation to our tissues. For this reason, neutrophils circulate in the bloodstream and are only called in by macrophages when they are needed. They are also very short-lived, only about 5 days. Finally, if an attack persists or is too overwhelming for the innate cells to handle on their own, it's time to call in the real cavalry in the form of the adaptive system. This is the job of dendritic cells. Dendritic cells also phagocytose pathogens, but instead of digesting them to get rid of them, they can use parts of them called antigens to induce an adaptive immune response. To do this, the dendritic cell regurgitates a small antigenic particle and presents it on its surface, then travels to nearby lymph nodes to activate T cells. Dendritic cells are therefore one of the aptly named antigen-presenting cells. Macrophages present antigen too. The difference is that they don't leave the tissue and can only activate cells that come to the front lines to help out. As we touched on earlier, the adaptive immune system works to boost innate defenses by producing antibody, and one of the jobs of antibody is to coat pathogen surfaces to help phagocytosis. All phagocytes therefore have receptors for the Fc portion of antibody, which helps them to feast on some otherwise resistant bacteria. The C3b complement protein works similarly to antibody in this regard, and phagocytes can recognize C3b as well. Lastly, there are a bunch of other innate cells we'll learn about, such as natural killer cells that protect us from viruses, and eosinophils that help defend us from parasites. Now that we've introduced the concept of antigen, let's further define it and see how it relates to the eventual production of antibody. An antigen is defined as any molecule, even belonging to our own body, which elicits an adaptive immune response, and more specifically, an antibody response. An important point here is that the antibodies created in response to a certain antigen are specific to that antigen, meaning they can only bind to something containing that antigen and nothing else. This allows the immune system to target only the bad stuff. Let's talk about how this happens. T and B lymphocytes have receptors that recognize antigens as either belonging to our body or as foreign. Each cell in its receptor can only recognize one antigen, referred to as its cognate antigen. The specific molecular pattern on an antigen that binds to and is recognized by the receptor is called the epitope, and that's what this graphic is showing. This is the antigen in the middle, and each antigen is comprised of smaller molecules on its surface to which antibodies can attach at their antigen binding region, and those are the epitopes. This part of the antibody down here is called the Fc region and allows antibody to be recognized by immune cells. When a lymphocyte receptor does locate and bind to its antigen, that's when the immune response can really take off, and it does this by clonal selection. Clonal selection is the preferential proliferation of only those specific lymphocytes that recognize their antigen. Therefore, you get the production of only that specific antibody we need to target that antigen and fight that particular infection. If you think about it, this is a pretty cool system. Our body can be exposed to just about any antigen, and so we must be capable of producing antibody to recognize every possible organic molecule that exists. It would be extremely inefficient for our body to carry around hundreds of thousands of lymphocytes for each and every antigen we might possibly be exposed to, just so we can fight that random infection if it happens to come up. So we have only a few of each type of lymphocyte and then mass produce them when the situation demands it. This accounts for the powerful and highly specific response of the adaptive immune system. Since lymphocytes can trigger a major immune reaction in response to antigen they encounter, they are taught early in their development and thereafter to differentiate between the enemy and self-antigen. This concept is known as tolerance and is in place to preclude the possibility of an autoimmune reaction. Let's take a break from this discussion for a minute and talk about the roles of lymphocytes in immunity. So pathogens that enter the body can be taken care of by cells like macrophages and neutrophils, but this is mainly true for your garden variety bug that is pretty easy to handle. Some bacteria, though, have developed mechanisms to evade capture by the innate system and require a much stronger counterattack. Also, the innate system is good at detecting pathogens outside of cells, but is limited in detecting intracellular bacteria and especially viruses. Viruses enter cells and use the host to replicate themselves. When they do this, they are for the most part safe from detection by immune cells outside of the cell they have infected. The adaptive system is much better equipped to handle these situations, especially in large infections, and leads to a more targeted response on a greater scale. And this response is actually broken down into two categories, or really two phases. The first is called cell-mediated immunity, and this is handled by T cells, and there are two main types of T cells. Cytotoxic T cells are cytotoxic, meaning they are toxic to cells. They directly target cells that are perceived as threatening to the body, such as those infected by viruses, and they knock them off individually. This is necessary because antibody cannot enter cells and only really works against extracellular pathogens. Cytotoxic T cells are known to be CD8 positive. Helper T cells express CD4. CD8 and CD4, as we will learn, are very important because they are co-receptors for T cell activation, and we'll talk more about this later on. Helper T cells get their name because they don't do anything directly to protect the body. When a helper T cell is activated by an antigen-presenting cell, it works to coordinate the immune system and sets it up in a way to best fight the type of pathogen infecting the body. It does this by secreting cytokines as signals to activate and induce changes in other immune cells. They can support cells of the innate system at the site of infection. They can activate cytotoxic T cells, leading to better cell-mediated immunity if there is a bad viral attack. But the way they really get things going is by activating B cells, especially when fighting resistant bacterial infections. This is their most important role because B cells are involved in the second level of adaptive immunity referred to as humoral immunity. There is a way that humoral immunity can be triggered without T cells, but for the most part, it exists in series with cell-mediated immunity, meaning T cells are usually a prerequisite to B cell activation. It's called humoral immunity, humor being the ancient term for bodily fluid, in this case blood plasma, because once properly activated by helper T cells, B cells can then differentiate into plasma cells. Plasma cells don't circulate, but rather secrete antibody into the blood, where it can then go on to enhance the innate system in a number of really important ways. Antibodies, otherwise known as immunoglobulins because they are essentially immune proteins, can take several forms or isotypes, which we call IgM, IgG, IgA, and IgE. Each has a slightly different function, but overall, like we said earlier, antibody are made to specifically target the infecting pathogen of interest and direct the immune response toward fighting it. Antibody doesn't kill anything but can tag a pathogen for destruction by innate immune cells or neutralize viruses by preventing them from entering cells or replicating. And like we have already stated, it does all this by binding the specific antigen associated with that pathogen. Antibody is actually a loose, non-anchored form of the B cell receptor and has the same specificity and affinity for the antigen that the B cell had. And this is a super important concept because one of the great things about the adaptive system is memory. Lymphocytes, when activated, start proliferating into the effector cells that take on the infection, but some of their energy goes into making memory cells as well. Memory B cells will maintain specificity toward the pathogen that triggered their formation, and if ever the body encounters it again, these cells are standing by to ramp up the adaptive system and produce antibody much more quickly and in greater quantity than the first time. Memory T cells can also be produced, although these are less well studied. This concept of immune memory is extremely important and forms the basis of vaccination, where we can prime the body to be ready for an infection before it has ever actually encountered it. So that's pretty much the immune system in a nutshell. The immune response at its peak uses antibody to help clear infections, and then will forevermore have a bunch of circulating lymphocytes that are on standby in case the same pathogen returns. The focus of the rest of this lecture will be to introduce certain concepts and the mechanisms of immune system activation necessary to the discussions we will be having later in the course. So first I want to get back to our discussion on the topic of antigen. An important aspect of the interaction between an antigen and the T cell receptor is that in order for T cells to recognize their cognate antigen, it must first be presented. Teleologically speaking, this was designed to help prevent T cells from being overreactive to our own tissues and causing autoimmune disease. Several cells have this capability to present antigen and activate T cells. These are the antigen-presenting cells we touched on earlier and include dendritic cells and macrophages, and also B cells. It makes sense for dendritic cells to present antigen as they can go to lymphatic tissue to activate the T cells that hang around there. Macrophages, on the other hand, don't leave the periphery, but we still need antigen presentation at the site of infection because activated T cells may go back to their resting state if they are not constantly being re-stimulated. B cells, like T cells, have receptors with a very high affinity for antigen. This allows B cells to concentrate antigen, which makes them pretty good antigen presenters. And this way, it's easy to activate T cells even when there's not so much antigen around. There are a few surface molecules that all antigen-presenting cells must express. The protein responsible for antigen presentation is called the major histocompatibility complex, or MHC. In antigen-presenting cells, it's MHC class II, and we will see why that is in a minute. B7 and CD40 expression are necessary for antigen-presenting cells to activate T cells and to be activated by T cells respectively, something we will get into a little more in our cell-mediated immunity lecture. Just know for now that somehow or another antigen-presenting cells, like pretty much all cells in the immune system, must be activated, either by the pathogen itself or by T cells which recognize that pathogen before they can go into antigen presentation mode. Let's continue our introduction to the immune system, staying on this very high-yield topic of MHC, which is central to antigen presentation, and is really important to get a handle on prior to delving deeper in the coming lectures. The major histocompatibility complex is a protein which is so called because it is used clinically when dealing with transplants to determine if donor tissue will be compatible with the recipient. Since the job of MHC is to bind T cells, it is extremely immunogenic and leads to transplant rejection if the MHC molecules don't match up. There are two classes of MHC that we will compare, and you will need to know the differences. The job of MHC class 1 is to give information to the immune system about what is happening inside a given cell. It does this by presenting intracellular antigen, and it does this all the time and for almost all our cells. This tells the immune system that everything is okay and our own cells aren't posing a threat. One of the differences they like you to know between MHC classes is the genetics. The genes for MHC are on chromosome 6 at the HLA loci. HLA stands for human leukocyte antigen, just another way scientists refer to MHC because it was discovered first to exist on white blood cells. The three MHC1 loci are called A, B, and C. You can easily remember this because they all have only one letter, for MHC1. Within each gene there are many polymorphisms, or subtypes, which are numbered, and so there are many possible combinations of these genes for any given individual. Certain autoimmune diseases have a known association with certain HLA subtypes. For example, someone can have the HLA-B27 subtype of the HLA-B gene, which has a well-known association with the seronegative spondyloarthropathies. Every person has two sets of these genes, one from each parent, and each set gets passed down in its entirety. Siblings then have a 25% chance of being perfect matches when being considered as transplant donors. Like we mentioned, if there is a large discrepancy in MHC genotype between an organ donor and the organ recipient, it can lead to tissue rejection. They therefore try to match these different loci as much as possible before they will attempt a transplant. In the grand scheme of things, the fact that there are so many different subtypes of MHC is a good thing, because if there was just one type, for example, a single virus that mutates to evade it could wipe out our entire population. MHC1 is found on all nucleated cells, including platelets, which are derived from a nucleated cell, but this is to the exclusion of red blood cells, which have no nucleus. If red blood cells did have MHC, it would make blood transfusions as tricky as organ donation. And just a reminder, MHC1 is expressed all the time to make sure the immune system knows everything going on in our cells is legit. Before antigen can be loaded onto MHC for presentation, it must first go through processing in the cell. The type of antigen that MHC can present is protein antigen exclusively. These proteins must get broken down into smaller peptide molecules before they will fit into the MHC binding groove. This allows for the detection of antigens on the interior of a pathogen and the activation of many different lymphocytes that would otherwise not have been exposed to their antigen. Let me diagram for you how this works to make it easier. Again, in normal non-infected cells, MHC1 presents antigen continually. Usually this is self-antigen, which ideally is not recognized by immune cells. Let's say this is an epithelial cell infected by a virus. In our cells, the ubiquitin proteasome pathway is generally responsible for protein catabolism. Older worn-out proteins, which I'm representing by this green triangle here, are tagged for destruction by the enzyme ubiquitin ligase and broken down to amino acids and peptides in a proteasome, which I'm drawing as this big orange circle. Some of these broken down parts are used to make new proteins, and some are brought to the rough endoplasmic reticulum by the transporter associated with antigen processing protein, or TAP, which also loads the peptides onto MHC. The MHC molecule is then sent to the cell surface to hold that peptide out or present it to passing immune cells. When the virus starts replicating and making its own proteins, those are also broken down, brought to the rough ER, and loaded onto MHC. When the MHC presents these foreign proteins on the cell surface, this can activate nearby cytotoxic T cells to destroy the infected cell. And MHC1 binds the T cell receptor with the help of the CD8 co-receptor. By the way, for visual ease, all T cells in this lecture series will be teal colored, teal for T cells, and I will be making B cells blue. So getting back to the text, I emphasize that proteins in cells, even ones made by viruses, are processed by the ubiquitin proteasome pathway, and this is important to know in general. And the special protein involved in MHC1 peptide loading is TAP. And once again, MHC1 presents intracellular antigen to cytotoxic T cells, which requires binding to CD8. And this makes sense because cytotoxic T cells are in charge of fighting intracellular viruses. And sometimes they want you to know the differences in protein structure between MHC classes. MHC class 1 has one long chain with an associated support protein called beta-2 microglobulin that is involved in antigen loading. Let's compare to MHC class 2. MHC2 presents exogenous antigen to communicate to cells what is happening in their outside environment, and it is only upregulated when there is infection. MHC2 loci have two letters. All of them start with HLA-D followed by P, Q, or R. And MHC2 is only found on antigen-presenting cells like we've mentioned. Antigen-presenting cells are in charge of expressing extracellular antigen to trigger T cells, and so must also be able to bring antigen into the cell somehow. Macrophages and dendritic cells are phagocytes and have no problem doing this. B cells, which are not phagocytes, bind their antigen using the B cell receptor, and the entire receptor-antigen complex can be enveloped in a process called receptor-mediated endocytosis. This system of bringing in and presenting extracellular antigen is necessary because many pathogens don't infect cells and stay outside them. Just like our own proteins, pathogens must be broken down into smaller molecules before their antigens can be presented. Let's go back to the whiteboard a minute. Let's say this is an antigen-presenting cell engulfing some kind of pathogen and breaking it down to its antigenic particles. Once inside the cell, a vesicle carrying antigen makes its way again to the rough endoplasmic reticulum where MHC class 2 is made. Here's our endoplasmic reticulum. Now let's just give it some ribosomes, which can help produce MHC2. A major difference from class 1 is that here, the antigen doesn't bind MHC2 just yet. A protein called the invariant chain sits in the binding groove, which prevents MHC class 2 from accidentally binding intracellular proteins. This is to safeguard from autoreactive T cells being presented with our own antigens. Once the MHC class 2 and extracellular antigen are packaged into an endosome and are safely away from the ER, the acid environment of the endosome causes the invariant chain to fall off, and the extracellular antigen can bind. MHC class 2 binds the T cell receptor with the help of CD4, so it can only bind helper T cells. By presenting helper T cells with outside antigen, it lets them know what kind of infection is taking place so that they can mount and direct the right type of response. A good mnemonic to help remember which MHC class binds which co-receptor is that when you multiply the MHC class and its associated CD molecule, you always get 8. So CD4 times MHC2 equals 8, and CD8 times MHC1 equals 8 as well. So getting back to our slide, we say that in antigen loading for MHC class 2 molecules, the invariant chain is responsible for preventing intracellular antigen from binding, and it's pretty high yield to know how it figures out when to detach, and it's due to an acidic reaction in the endosome. And of course, MHC2 binds CD4 only. And just for comparison to MHC class 1, MHC2 has 2 chains of equal length and no other associated protein. Again, the overall concept of MHC is super high yield, and that's why I've spent so much time on it and highlighted a whole bunch of stuff in red. Now that we have a good foundation regarding the cells of the immune system and how they function, and before we go into more detail in the coming lectures, let's take a step back to the macroscopic level to talk about the different organs involved in the immune system. We'll take a closer look at each one on a tissue level in their respective lectures. Organs of the immune system are broken down into 2 groups. Primary immune organs are the organs where immune cells mature, not where they're made, where they mature. Like we already talked about, all blood cells originate from bone marrow. However, T cells leave the marrow and mature in the thymus. B cells, however, stick around in the marrow and continue their maturation process there. After maturation, lymphocytes hang out in the secondary lymphoid organs, and this is where antigen recognition takes place. All of the following tissues contain lymphoid follicles, where B cells can proliferate in response to infection after exposure to their cognate antigen. The most abundant of these tissues are the lymph nodes. Lymph nodes are distributed throughout the body to make sure there are enough cells around for an attack at a specific site. Lymphocytes go from node to node looking for antigen and complete an entire circuit within a day. This effectively increases the probability that the adaptive immune system will be activated. The spleen is a larger lymphoid organ in the abdomen and has several other functions in addition to its role in immunity. and Wilder's ring of tonsillar tissue protects the airway and gut from inhaled and ingested pathogens. The previous three organs all have a capsule surrounding their lymphoid tissue, but there are also these entities called mucosa-associated lymphoid tissue, or MALT, which are scattered throughout the gut to protect against foodborne pathogens and are unencapsulated. Included in these are the so-called Peyer's patches found in the ileum of the small intestine. One of the ways for immune cells and antigen to get from tissues to these organs is through the lymphatics. Plasma leaks out of blood vessels into the interstitium when the hydrostatic pressure of blood is high or the interstitial pressure is low. It can then return to the circulation through small lymphatic channels. Since this is a very low-pressure system, lymph is worked along mostly by muscle contraction and takes immune cells with it. Lymph drains through these channels and into lymph nodes, which are distributed all throughout the body at strategic junctures associated with specific tissues. It's good to know which draining lymph nodes are from which area of the body to be able to predict where an infection or a cancer originated, and this usually follows the blood supply. After leaving the lymph node, lymph goes back to the circulation through efferent lymphatics, which all merge and drain into the thoracic duct, a lengthy structure that runs behind the aorta and is highlighted in green in this image. This duct carries all the lymph from the lower half of the body and the lymph from the left upper half of the body and dumps it all back into the circulation at the junction of the left IJ and the left subclavian veins right here. The right lymphatic duct carries lymph from the right upper half of the body and empties it into the corresponding junction on the right. This is how T and B lymphocytes circulate throughout the body and are able to make multiple passes through many lymph nodes in search for their cognate antigen. Now that we've seen the big picture, let's go to the whiteboard for a moment for a schematic of the immune system to really get it down. Let's say the body is attacked by a virus or bacteria or really anything, fungus, parasite, you name it. Your innate physical barriers, such as your skin, has macrophages there to recognize this and will send out cytokine signals to trigger acute inflammation. And neutrophils will come in to help destroy the pathogen at the site of attack. The innate system is perfectly capable of handling small-scale infections, especially when they are viral or fungal. For larger or more virulent infections, if the battle goes on long enough, dendritic cells have time to engulf some of the pathogen and travel through the lymphatic system to lymphatic tissue, such as this lymph node, and present antigen to helper T cells there. When the helper T cells recognize their antigen and are activated, they now direct the rest of the immune system to fight back accordingly. They can release cytokines to enhance the activity of innate cells like macrophages. In a bad viral attack, they stimulate cytotoxic T cells, and in bacterial infections, they initiate the humoral B cell response. B cells differentiate into plasma cells, which secrete antibodies specific for their antigen, and which can latch onto the pathogen, making it harder for them to invade and easier for phagocytes to kill them. After an infection is cleared, most lymphocytes die. We don't need a bunch of extra cells lying around with no purpose. But B cells will also make a number of memory cells that will hang out for a long time after, ready to fight that same infection if it happens to return. The last thing I would like to do in this introductory lecture is to take a broad look at immunodeficiency. I will be going through all the different immunodeficiency syndromes as they come up in their associated lectures, but I wanted you to have a general idea as to what kinds of infections might crop up when missing any of the components of the immune system, because when reading a boards passage, you'll be better equipped to figure out how to categorize the problem even if you don't remember the name of the specific disorder. If someone has a granulocyte deficiency or neutrophil dysfunction, they will not be able to handle common bugs found in the periphery, namely the skin and soft tissues. So you will see things like frequent infections with common bugs, for example, staph abscesses. Also, in a patient with innate system dysfunction, you will find fungal infections. Candida especially is known to be captured and handled by nets, neutrophil extracellular traps. Without proper neutrophil function, Candida can get into the blood, causing systemic candidemia. This is not uncommon in patients receiving chemotherapy who tend to be neutropenic. With T cell dysfunction, remember that cytotoxic T cells help out with viral infections. So think about some of those nasty viruses immunosuppressed patients can get, such as CMV, EBV, zoster, and other chronic respiratory and GI viruses. Cell-mediated immunity also helps the innate system deal with fungal infections, including some of those opportunistic infections like pneumocystis or crypto that you can see in AIDS patients, whom essentially are T-cell deficient. T-cell immunity is very involved in the fight against Candida as well, but this is limited to local Candida infections. This is why people with AIDS and asthmatics inhaling corticosteroids are susceptible to getting oral thrush, and I've included a photo of what that looks like. The role of T-cells in the local reaction to Candida is also the basis for the Candida extract skin test for T-cell function. But remember, as long as neutrophils are present, Candida will not get to the bloodstream. So T-cell dysfunction equals local Candida, neutrophil dysfunction, systemic Candida. Lastly, B-cell dysfunction typically results in recurrent bacterial infection, B for bacterial, because antibody is needed to assist in the killing of many bacteria that have a resistant capsule. The ones most frequently seen are strep pneumo and Haemophilus, which also happen to be the most common bugs involved in sinorespiratory infections. People lacking B cells will keep getting the same infections because they don't produce a memory antibody response either. So in a passage, if you have a patient with a history of recurrent ear infections, sinus infections, pneumonia, or whatever, for the boards, 90% of the time that is code for some kind of B cell deficiency. The antibody immunoglobulin A is especially important in dealing with Giardia, a gut parasite, so B-cell deficient patients may also have episodes of diarrhea in their history. Another function of antibody is preventing viruses from entering cells, so these patients can get certain viruses, like enterovirus. And don't forget that T cells are needed for much of the humoral immune response, so certain T cell deficiencies can result in a quote-unquote combined deficiency, where patients will also have some level of B cell dysfunction. This can make diagnoses more confusing, but if the only history they give you is recurrent bacterial infection, they are likely looking for a pure B-cell dysfunction. So that's it for my immune system introduction. In the next 3 lectures, we will take a more in-depth look first at the innate system, followed by cell-mediated and humoral immunity. If you like this video, share with your classmates and friends, give it a big thumbs up, and hit the pathognomonic icon to subscribe to the channel. And we'll be coming out with more videos on other high-yield boards topics, so ring the bell if you'd like notifications. And as always, please leave your comments below and tell us what we're doing well and where we can improve. Thanks for watching, and I'll see you next time.